evening, everyone. Emory Moynagh here for Creation Biology, and we're going to be reading Kurt Wise's paper from 1990, the foundational paper in Baraminology. Its title is Baraminology, a Young Earth Creation Biosystematic Method. I read Walter Remind's paper last week. I'll be reading this one this week. This one's going to be a little bit longer. It's about 17 pages. But hopefully we can get through the whole thing tonight in one sitting. Lord willing, let's fingers crossed. That's what we're going to try for. I'm also going to be reading Randy Galuza's Continuous Environmental Tracking paper. Unfortunately, that paper is significantly longer. There are a few figures in there, but it is a significantly longer paper, so that one may take 2 videos, but that's in the plans, at least for the moment. We'll see how we do when we get to that point. But for the moment, we're going to go ahead and read Kurt Wise's paper on Baraminology, a Young Earth Creation Biosystematic Method. By the way, if there is a paper within creation biology you want me to read as part of the podcast and the discussion, feel free to let me know about that in the comments, and I'll be happy to if I can. Now, certain topics are beyond my expertise. If it's a protein biology paper, you might want to be better off taking it to Sal. But if it's something related to genetics, baraminology, diversification, that sort of thing, that might be something more up my alley. Right, let's get to the abstract. Walter Remind's discontinuity systematics can be used as a basis for a biological classification system, either within creation or evolution theory. Such a model-neutral methodology can be more efficiently utilized within any given theory of life by supplementing it with model-dependent membership criterion. Discontinuity systematics supplemented with young Earth creation model-dependent membership criterion is here called baraminology. 4 young Earth creation model-dependent membership criterion are here suggested. 1 based on scripture, another based on molecular similarity studies, a third based on cladistically defined frequency of homoplasy, and a 4th based on flood-generated diversity bottlenecks. Okay, there's a lot in that paragraph, but he's going to talk about all those things later. So we'll come back to them, okay? This paper also attempts to relate the empirically defined holobaramins, monobaramins, apobaramins, and polybaramins of baraminology and discontinuity systematics to biblical, creationist, and evolutionist thought. The original created group of individuals capable of reproduction is a theoretical construct, here called an archibaramin. Frank Marsh's term baramin is considered the term created kind, or biblical kind, and is here redefined as the archibaramin and all its descendants. Baramins are apobaraminic groups and may be holobaramic in most, if not all, cases. Microevolutionary processes, including speciation, occur within the baramin. Extra and interbaraminic evolution is considered impossible. So again, a lot there. We'll come back to all this as we go on. I'm just reading you the abstract, and then we'll get into the information. All right? Baraminology is easily employed and extremely powerful biosystematic method. Baraminology is the most efficient method of classifying life available to young Earth creation biologists. It will allow the collection of an abundance of heretofore unrecognizable data in favor of a creation model and serve as an empirical foundation upon which to construct a creationist reclassification of life. And boy, I wish all that was true. It's turned into more of let's see how much of evolution we can shove into creationism. Anyways, that's beyond the scope of probably what we're going to get into here. Introduction. 18th century creation theory viewed life as both invariant and typological, both unchanging and polyphyletic. This view can be described as a creationist lawn view of life, see figure 1a, which I will hopefully be able to put up on the screen. In this metaphor, each species in the creationist Earth's biota is represented by a distinct blade of grass in a newly cut lawn. Each blade has its own separate root or origin and continues upward through time unchanged, as one sprig of grass among many thousands. Darwin challenged the creationist lawn metaphor with a metaphor of his own, the evolutionary tree, see figure 1b. In this metaphor, life is monophyletic, i.e. there is one common ancestor at the base of the tree of life. As one follows the trunk upward, it branches repeatedly to produce the many thousands of terrestrial terminal twigs we recognize as species today. Evolutionary theory thus views life as monophyletic, variant, and relatively unconstrained. By the beginning of this century, Darwinian's evolutionary tree had largely displaced the previous metaphor of the creationist lawn. Note that he's writing in 1990, probably wrote the paper in 1989, so when he says this century, he means the 1900s, not the 2000s. Just piece of information there. And he's substantially correct in what he just said in that paragraph. Most people believed in fixity of species in Darwin's time. And Darwin did upend that particular apple cart, with good reason, I might add. But Darwin, unfortunately, took it a whole lot further than that, and that's the problem. Next paragraph. Some modern creationists are suggesting a metaphor of their own, a metaphor which is planted somewhere between the evolutionary tree and the creationist lawn. The new metaphor may be described as the neo-creationist orchard, see figure 1c. In this metaphor, life is especially created, as fruit trees are specially planted, and polyphyletic, i.e. each tree has a separate trunk and root system. There are also discontinuities between the major groups. Trees are constraints to change. Trees are spaced so that branches do not overlap and could not, and never did, anastomose. And there are constraints to change. A given tree is limited to a particular size and branching style according to its type. In these ways, the neo-creationist orchard is similar to the creationist lawn. They differ, though, in that the neo-creationist orchard allows change, including speciation, within each created group. Each tree branches off of the main stem. Permitting this kind of change, variously called by creationists as diversification, variation, horizontal evolution, and microevolution, in different amounts in different groups, allows the creation model to accommodate microevolutionary evidences, e.g. changing of allelic ratios, genetic recombination, speciation, etc. And again, this is substantially true, although I will say that Rob Carter has put together an article series on the braided baranome, which I think is worth reading at some point. I plan to read it at some point, Carter's article series. I think it's very interesting, which may eventually supplant the neo-creationist orchard, and probably, maybe should, just as a bit of extra information, but we'll get to that another time. We're only talking about Wise's work. And again, this paper is 30 years old, so I don't expect everything that is said today to match up to a 30-year-old paper, but it's important to read this paper because it gives you foundation for what's to come later on in creation biology. According to the creation model, genetic and morphological discontinuities are not only extant, but they are also a common and important characteristic of life. To be consistent with their own model of Earth history, creationists will need to identify discontinuities among life forms, then use them to classify those life forms. Unfortunately, all of the traditional biosystematic methods are incapable of either recognizing or utilizing any such discontinuities, even if they were a common characteristic of life. Phylogenetic systematics and evolutionary taxonomy explicitly assume continuity, and thus always conclude that continuity is a characteristic of life. The methods of phonetics and transform cladistics are also blind to discontinuities for three reasons. First, each method orders organisms only according to similarities. Differences are employed only in the sense of dissimilar similarities, e.g. synapomorphies or shared differences. Since similarities can be identified between any two items in the universe, it is impossible to identify any discontinuity using these methods. That is very, very important, because the evolutionists like to back away from the young Earth creationists who are in the Baraminology camp. They like to back away from cladistics, and they say that, oh well, we don't actually do anything with statistics. Statistical Baraminology isn't cladistics. It just detects continuity and discontinuity. Well, no, it doesn't detect discontinuity. All it does is detect continuity, and you presume that discontinuity exists based on the existence of continuity, because the Beatist algorithm is drawn directly from phonetics. Robinson and Kavanaugh admitted as much in the 1998 paper, or 96 paper rather, where they introduced the method. Go read it for yourself. I think it's available online. If it's not, I can probably get you a copy. They admit it. So it is drawn from phonetics. And we're going to get back to that later on, because Wise is more than happy to use cladistics and phonetics for his particular purposes. Continuing on. Second, the products of these methods are data structures such as phenograms and cladograms. These structures locate life forms at the tips of branches of tree-like diagrams. By their very nature, such diagrams connect all organisms considered, so it is not only impossible to identify discontinuity, but it is also impossible to graphically display it. That's true. Third, phenograms and cladograms are so similar to the representations of evolutionary trees that many people wrongly conclude that they do represent evolutionary phylogenies. The very appearance of these data structures persuasively but subtly argues for the inherent continuity of life and against discontinuity. All traditional biosystematic methods, because they are insensitive, completely blind to discontinuity, are thus inappropriate classification methods for creationists who are seeking to find the discontinuities they believe to characterize life. And this, by the way, I think is very good thinking, and this is where he's borrowing from Remind. Now, it's interesting that he says here that these methods are inappropriate to classify life, but yet will go on later to recommend using them. Like, that I don't completely understand, but we'll come back to that later. Next paragraph. Walter reminds recent introduction of discontinuity systematics should be very encouraging to creation biologists. Discontinuity systematics can be used to identify the discontinuities of life, which creation theory predicts, and then use that information to classify life. It is also very simple conceptually and is easily modified to the creation model. After briefly familiarizing the reader with discontinuity systematics, this paper will introduce a modification for use in young Earth creation theory. So Kurt Wise is literally ripping off, with permission probably from Remind. I'm not saying he's stealing it. I'm not saying he's dishonest, but he's taking discontinuity systematics and trying to adapt it to creation science. And I think he did so relatively poorly. But we'll continue that as we go on. Header discontinuity systematics justification. Two organisms, or organismal groups which appear to lack a common ancestor, can be said to be separated by what might be called a phyletic discontinuity. A group of organisms which contains creatures related to one another but unrelated to organisms outside the group makes a truly natural group. Since biosystematic methods search for natural groups and classify organisms accordingly, phyletic discontinuities can be used as a basis for classification. Remind's discontinuity systematics does just that. It focuses on searching for and identifying phyletic discontinuities and then using them to classify organisms, which is true. One of the strengths of discontinuity systematics is that phyletic discontinuities do not necessarily have to be real for the classification method to work. Since our knowledge of the world is partial, identified phyletic discontinuities can be either real or apparent. And by apparent, by the way, he means that we could be perceiving them because we don't have enough information. Some, many, or all of the phyletic discontinuities we identify may be separating organisms which really do share a common ancestor. It is still possible, for example, that different mechanisms of genetic change separate different organismal groups. If so, these thresholds of change can still be used to classify organisms into natural groups. Another strength of discontinuity systematics is that as current research changes the nature and position of phyletic discontinuities, discontinuity systematics can accommodate those changes. A third strength is that discontinuity systematics is easily adapted to different theories of life. Macroevolutionary biologists, for example, assume that all phyletic discontinuities are apparent until it is demonstrated that they are real. Currently recognized discontinuities may well reveal the existence of a mechanism to change yet not characterized, e.g. macromutation, regulatory gene mutation, etc. To conclude that a phyletic discontinuity was real would be the last choice of a macroevolutionary biologist. Creation biologists, on the other hand, believe that all life is polyphyletic. They would assume that phyletic discontinuities are real until they are demonstrated otherwise. In some small way, therefore, the creationist would tend to interpret the phyletic data more literally than the evolutionist. So what he's effectively saying here is that the evolutionists assume that similarity equals ancestry until it doesn't, and so basically until the point where even if there's a discontinuity, there's still some ancestry until you can prove otherwise. Whereas creationists say, no, the discontinuities are real. Now, I'm not opposed to discontinuities. As a matter of fact, I like discontinuity systematics as an idea. But I do think the way it's been implemented in statistical baraminology is rife with problems, and that's a whole other kettle of fish we'll go down later. Discontinuity systematics involves the identification and classification of organismal groups completely bounded by phyletic discontinuity. Remind felt that most of the biosystematic terms in current usage are inappropriate to describe this kind of biological group. Because species is derived from the Latin word for kind, the divinely created kind, it would seem to be appropriate for this unit. Yet, even though etymologically species is used to describe a group of organisms completely bounded by phyletic discontinuity, nowhere in current usage does the word species carry that meaning. It is unreasonable to think that a new meaning could be added to the word species without causing undue confusion. A second word is often used by creationists, kind. This has a reference to the created unit. However, kind also has too many colloquial meanings to avoid the inevitable confusion that would come about with the use of this word. As a result, Remind turned to the word baramin, created by Frank Marsh. A combined form of two Hebrew words meaning created kind, Marsh envisioned the baramin as seeing both the created biological unit and the reproductive unit. Remind used this word as a root word for the creation of the terminology of discontinuity systematics. Wisely, Remind did not use baramin itself to mean anything in his system. This allows the terminology of discontinuity systematics to be model neutral. Clark's baramin can continue to refer to the created biological unit and still allow Remind's terms to exist independently without a creation model-dependent meaning. Now, maybe they were more hopeful of rapprochement with the evolutionists in 1990, but I really don't care about this. Like, I'm not entirely sure of having a systematic system that could fit with the evolutionists and the creationists. I don't understand why you would even care about that. Evolution's wrong, creation's right. So why are we not building our models based around that instead of trying to build something that's model neutral? I mean, the evolutionists are never going to give us rapprochement, so I don't know why we would try to build our models that way. But again, this is 1990. This is 30 years ago. Maybe both Hermione and Wise have figured this out by now. I don't think Wise has. Maybe Hermione has. I don't know. 4 terms were introduced by Remind to create the necessary terminology for discontinuity systematics: Holobaramin, Apobaramin, Polybaramin, and Monobaramin. Each of these terms is used to describe a set of known organisms. Since this is a method of classification, it is only used to describe and classify organisms which are known to exist or to have existed. It does not include imaginary creatures. The Holobaramin is the desired basic unit. This is a group of organisms which is surrounded by a phyletic discontinuity, and yet is not completely divided by one. Once all the holobaramins and phyletic discontinuities have been identified and characterized, the primary goals of discontinuity systematics will have been achieved. The monobaramin is a group of organisms which is not completely divided by a phyletic discontinuity, but may or may not be separated from all other organisms by phyletic discontinuities. In other words, the monobaramin is a subset of the holobaramin. The polybaramin is a group of organisms divided by at least one phyletic discontinuity. The polybaramin may or may not be completely separated from all other organisms by phyletic discontinuities. Thus, the polybaramin contains at least parts of at least 2 holobaramins. So, effectively, a polybaramin has 2 or more holobaramin pieces contained in it, which are not from the same holobaramin. The apobaramin is separated from all other organisms by phyletic discontinuity, but may or may not be divided by at least one phyletic discontinuity. The apobaramin thus contains 1 or more complete holobaramins. Remind's actual definitions are included in Appendix A. I read those in the last podcast. We're not going to read those again. These terms are pluralized by the addition of an S, e.g. 5 apobaramins and 6 polybaramins, and are made into an adjective form by adding ic, e.g. reptiles are polybaraminic and maybe apobaraminic. Again, that's all review from Remind. If you watch the Remind podcast, you've gotten all this information. Methodology of discontinuity systematics simply involves identifying holobaramins through a method of successive approximations from what might be termed above and below, by successive subtraction and addition respectively. Holobaramins are approached by addition by identifying monobaramins and increasing their membership. In this way, the holobaramin is approached from below. Holobaramins are approached by subtraction by identifying apobaramins and dividing them into smaller apobaramins along identified phyletic discontinuities. In this way, the holobaramin is approached from above. Approach the holobaramins simultaneously from above and below by successive subtraction and addition allows the systematist to identify the holobaramin most quickly. Again, all this is review from Remind. And I am going to jump a little bit ahead because we are going to skip over the rest of the review from Remind because we've done that podcast. If you want to go review that, you can go watch the podcast I did on Walter Remind's paper, and we're 18 minutes in. This paper's 17 pages long. I want to jump ahead. So we're going to jump ahead to where he starts talking about baraminology. You can read the rest of the paper. I will link to it. It is perfectly fine for you to read that, and I suggest you do. But we're going to jump ahead to Remind's paper. So to the actual part that Wise is new, because, I mean, so far Wise hasn't really told us anything that was new. He's just reviewed discontinuity systematics. Baraminology. This is on page 348 of the Proceedings of the ICC. Justification. Although discontinuity systematics is useful because of its adaptability, it is not the most efficient method of identifying phyletic discontinuities and classifying organisms. Depending upon a researcher's view of life and its history, membership criterion can be added to those of discontinuity systematics to make the resultant biosystematic method more efficient. Since, however, this method will be dependent upon a particular model for the history of life, this biosystematic method will be model-dependent. To maximize the efficiency of their biological classification, researchers employing any particular model of life's history should adapt discontinuity systematics to their particular model. The author here is modifying discontinuity systematics by adding membership criterion, which are young Earth creation model dependent. The resultant biosystematic method here is called baraminology. Alright, so now he's saying, okay, I've got discontinuity systematics, that's the foundation, that's what I want to build on. Here's what we're going to steal, here's what we're going to add in. We've already borrowed the things like similarity, successful hybridization, known variation, unambiguous lineage, and discontinuity. All those things come from discontinuity systematics. Now he's adding new stuff with some new terms. Baraminology uses all the terminology and methodology of discontinuity systematics. However, since it is based upon a specific model of the history of life, Baraminology often claims a particular interpretation of discontinuity systematic terms. Because young Earth creationism maintains that many different groups of organisms were separately created, it posits that life is polyphyletic and full of actual phyletic discontinuities. Whereas discontinuity systematics studies discontinuity, it makes no attempt to determine whether the phyletic discontinuities are real or apparent, to determine why they exist. Baraminology, on the other hand, claims that the phyletic discontinuities are real and due to the polyphyletic origin of life at the hand of a creator. So he's drawing a distinction. And this is one thing I do like about this paper, even though I don't really like a lot of where Baraminology has ended up. I do like this about the paper. Wise is deliberately trying to build something that fits the creation model. I don't know how well he necessarily succeeded, but at least he's not completely being model independent here. I think this is actually worthwhile. He's trying to build something that fits the biblical model. Membership criteria. Criteria of discontinuity systematics. Baraminology accepts all the continuity, additive, membership criterion thus far proposed for discontinuity systematics and the rationale for those criteria. Again, you can review that by watching my podcast on Remind's paper. Baraminology supplements those criteria with criteria based upon the young Earth creation model. Although the proposed discontinuity criterion of discontinuity systematics, substantial failure to show phyletic continuity, is also accepted, it is suggested that whenever possible it is not to be used alone. If additive and subtractive approaches to holobaramins are to be the most effective and error-free, the continuity and discontinuity criteria would be best defined independently of one another. If it is at all possible, neither should be defined simply as the negative of the other, particularly when one is defined as the failure to get positive results using the other. Such negative evidence, if it is all the evidence you have, is adequate for the moment, but systematists should be encouraged to find stronger, positive ways to define a discontinuity. The discontinuity criterion of discontinuity systematics, though such a negative criterion, is the only one so far defined. Baraminologists are encouraged to adopt positive discontinuity criteria to strengthen their argument for discontinuity. Several such criteria are suggested below. Now, I want to point out something here. The reason that hybrid data was effectively dumped out of statistical baraminology. I don't see very many people using it anymore. Admittedly, mostly because a lot of people were working with fossil data sets. But it mostly dumped out of baraminology was because it was only an additive criterion. You couldn't really subtract with it. That was the reasoning that Todd Wood gave when he wrote about it. Well, here, Wise is saying that discontinuity is only a subtractive systematic. It's only a subtractive measure. And that says something about the current state of baraminology, because continuity and discontinuity within the model are measured using are flip sides of each other. You can be continuous, non-continuous, or discontinuous, but they're measured using the same metric. In other words, if you're not completely similar, if you're not similar enough, you're dissimilar. So they're using them as flip sides of each other, which is what he argues against here. So it's very interesting how statistical baraminology has changed since Wise first put the word baraminology into the creationist lexicon back in 1990. And probably if Wise was being honest and still agrees with this position, he would say that that's incorrect and you shouldn't be doing that. Biblical criterion for discontinuity. Since most advocates of young Earth creation model accept the authority of scriptures, it is here suggested that baraminology should include, or at least consider, biblical criterion for the definition of holobaramin and apobaramin. In actual fact, the scripture offers only a very few suggestions, but they may turn out to be very important in the early development of baraminology's membership criterion. They can be used in some cases as a check on the reliability and or adequacy of other membership criterion. Folks, this is never done anymore. This was good, but it's gone off the rails. Since man is separately created, Genesis 1:26 through 27 and 2:7, etc., and all people have descended from Adam, Genesis 2:21-23 and 3:20, humans are holobaraminic. Similarly, on scriptural grounds, the tree of the knowledge of good and evil, Genesis 2:9 and 17, and the tree of life, Genesis 2:9, 3:24, each constituted its own holobaramin, each presumably having gone extinct after having been represented by only a single individual. The serpent also, Genesis 3:1, and whatever it became, Genesis 3:14, constitutes its own holobaramon. It is not impossible that it, too, became extinct with the death of a single member. That's interesting speculation. I don't know that I would have applied holobaramons to the Tree of the Knowledge of Good and Evil and the Tree of Life, given that those are not natural things. Those are supernatural things, so I don't know that I would have applied holobaramic status to those. But it's a nitpick. It's not something serious. It's kind of interesting that he does that. I'm not entirely sure why, but that's interesting. All right. Scripture also strongly implies that the raven, Genesis 8:7, and the dove, Genesis 8 through 12, must be separated by a real phyletic discontinuity and reside in separate holobaramons. Since the 2 or 7 of each land baramon were taken onto the ark, their separate mention indicates that the dove and raven are from different holobaramons, and in turn must be separated by a real phyletic discontinuity. We can also infer that the land plants, the sea and winged creatures, and the land animals are 3 separate apobaramons, since each were created on separate days of creation week, Genesis 1:11 through 13, 20 to 23, 24 to 31. The land plant and land animal creatures and winged creatures are similarly separated by phyletic discontinuity, Genesis 1:20 to 21. We might also infer that the thorn-bearing plants make up yet another apobaramon within the land plant apobaramon. The organisms listed elsewhere in Scripture, especially those listed in the dietary laws, may further aid us in establishing groups according to the biblical criterion, e.g., cud-eating mammals as a separate apobaramon. Now, I want to point out a couple things here. First of all, I appreciate the fact he's going back to Scripture. That's so rarely done in baraminology anymore, so I'm very, very thankful for that. Second, I have concerns about using the dietary law list, and I've enumerated those elsewhere, so I'm not going to go into a full detail with that, but I don't think the dietary law list is a good list for created kinds. It might be useful for apobaramons, but not for created kinds, for the very simple reason that the word kind did not necessarily have the same meaning in Exodus as it does in Genesis. In Genesis, the context makes the meaning clear. In Exodus, the context can also make the meaning clear. And we can go into that later if you'd all like. Much work can still be done in the original languages of the Scriptures to give us further clues about the higher classification of life. It must be cautioned, however, that Scripture provides only a very few clues about the definition of baramons. It remains for creation systematists to do much research to fill in the many gaps. Another caution is that most of these designations are themselves somewhat tentative. For example, we often do not know for sure to which organism a particular Hebrew word is actually referring. As a further example, there is uncertainty over exactly how the 3 groups of plants and the 3 groups of land animals in Genesis 1 are defined. We can use Scripture as an initial springboard, but it's probably only rarely that it will provide us with any definitive answers. Once defined and clarified, however, these biblical criterion may be very important in evaluating other criteria of baraminology. For example, the knowledge that humans are holobaraminic makes it clear that morphological and genetic similarity is insufficient evidence for phyletic continuity for humans and chimpanzees, are very similar in morphology and structural DNA. No and no. That's false on both counts, but that's neither here nor there. Chimps are probably most like us of all the animals, but it's very easy to tell a human and a chimp apart. And it's the same is true of the DNA. The DNA sequences are similar, but they're not that similar. The 99% number or whatever is a lie. Furthermore, until a criterion is located, in addition to the failure to bridge the morphological gap, which can distinguish humans and chimpanzees, then we know that other criterion are still to be identified. That's interesting. He seems to be admitting that we can't distinguish humans and chimpanzees right there, which is true in statistical baraminology. You can't, but, and I think there's an article on the IG website that demonstrates this relatively well. I think it's, what's his name? Somebody wrote an article on this explaining why baraminology is a dumpster fire. It can't separate apes and humans, but we'll go into that maybe more later as well. Maybe we'll read that as well. Molecular typology. Now this one is another one. This is DNA sequencing. Statistical baraminology threw out DNA sequencing a long time ago and hasn't really used it since. Occasionally, on very rare occasions, it gets used. Most of the time, it's purely morphological data sets, which admittedly for fossil data sets I understand, but it's not always just fossil data sets. Molecular typology. Comparative DNA studies have recently yielded some very interesting results. In these measures of similarity, the similarity between a given organism and a number of others seems to assume various high values for a certain small set of other organisms, whereas the similarity with any other member of the Earth's biota seems to assume a somewhat invariable non-zero value. This is true, for example, when archaeobacteria are compared with other organisms. The similarity among species of archaeobacteria examined is in a range of high positive values, but the similarity between archaeobacteria species examined and any given non-archaeobacterium species examined is virtually the same. This may be due to the fact that archaeobacteria form an apobaraman. Similar sorts of molecular typology examples can be seen in Denton. I'm assuming that's Michael Denton. It is suggested that using ANOVA and related statistics on molecular similarity matrices can be used to define coherent apobaramic groups. It can be postulated that the group which has significantly similar similarity values, which are also equally dissimilar from another organism, is separated from that organism by a phyletic discontinuity. Now, that's not a half-bad idea, and ANOVA was brought in briefly, and I say briefly, it's only got used a handful of times. It doesn't get used very much, and I know of no public application that allows people to use it if they have a data set. That said, if you wanted to go down this road, I think this is something that could be done, especially since we have much better DNA data 30 years on from this, but it has not been done as yet. I'd like to see more of it, though, so this is not a half-bad idea. It is suggested that baraminology provisionally accept the statistically determined discontinuities of molecular similarity as evidence of phyletic discontinuity. Baraminology also welcomes the different divisions made by different molecules as a way to maximize the efficiency of apobaramin construction. What one molecule divides into two apobaramins, for example, another molecule might divide into two different apobaramins. Using both molecules, it may then be possible to create 3 or 4 apobaramins. Using many molecules on many different organisms, the efficiency of baraminology should increase. Again, this has not been done, and I'd kind of like to see it done. I'd like to see a bunch of different genes. I think Gordon Wilson did a paper, over a year ago now, where he talked about orphan genes, using orphan genes and differences in genes to as ways to discuss the baramin. I think that would be worthwhile at some point potentially. Now, this next one is frequency of homoplasy, and he spends quite a bit of ink on frequency of homoplasy. So we're going to go over frequency of homoplasy fairly thoroughly here. Frequency of homoplasy, I do want to point out, this is kind of a cladistics idea, and we'll get more into that as we go further along, but frequency of homoplasy does borrow somewhat from the cladistic evolutionary classification system. If biblical claims are correct, then organisms can be divided into a number of separately created organismal groups, baramins. All genetic and morphological similarities between organisms of different baramins would then be homoplasias, i.e. non-inherited similarities. That would be true. Homoplasias similarities, then, would be a common feature of life. In fact, creationists have insisted for some time that the common hand of the creator would produce many similarities among unrelated organisms, e.g. Ascites from 1879. That's Louis Ascites, if I remember correctly. Agassiz, I'm not sure how to pronounce that. I think it's French. Since there are most probably many baramins and many more interbaraminic similarities per baramin than are currently known, young Earth creation theory predicts that homoplasy is an extremely common phenomenon of life. That's, by the way, true. Convergent evolution is a measure of homoplasy. Unlike the source of intrabaraminic diversity, diversity within a holobaramin is due to natural biological processes, including evolution. 30 years ago. It's right there in front of us. 30 years ago, and nobody saw it. Nobody noticed it. I guess it depends on what he means by evolution. What does he mean? It's not clear. In general, natural biological processes more easily produce similarity by common descent than by independent evolution. Again, what does that word mean? Does it mean change over time? Does it mean change within allele frequencies in a population over time? Does it mean from goo to you by way of the zoo over time? Doesn't say. We don't know. Therefore, within genetically related organisms, most of the similarities should be homologous in nature, i.e. should it be inherited from a common ancestor. This is true within holobaramins. This is true. However, the methods that are used to determine holobaramins, all similarity is the same. Similarity equals relationship in some way, shape, or form, according to the current methods of statistical baraminology, which is not what Wise is arguing here. He's arguing that a lot of similarities are homoplagious rather than homologous. But the current methods of baraminology assume that all similarities are homologous. They're identical to the way the evolutionists think. Whereas between holobaramins, the frequency of homoplasies should abound, within holobaramins, the frequency of homologies should abound. So in other words, between holobaramins, you should have a load of homoplasies. Within holobaramins, you should have a load of homology. But how do you determine one with respect to the other? That's the problem. And the thing is, you don't. The evolutionists, when they determine what a homology is, it's determined by reference to a phylogeny. So they determine how closely related something is and say, okay, these two traits are homologous. Or then they say, well, this trait over here is not defined by relationship, so it's not homologous, it's homoplagious. They're assuming what they're trying to prove. And Wise does not separate homologies from homoplasies in that sense. He assumes that the evolutionary definition of a homology is correct, and you can identify them based on their ancestry. Well, the problem is you can't identify a homology a priori unless you know what it's related to. You can only define it post hoc rather than pre hoc, if you will. You can't do it a priori, it has to be post hoc. But if it's post hoc, it's no help to you trying to determine relationships because relationships have already been determined. That's a huge problem, and that is probably the biggest underlying flaw in statistical baraminology, is that they assume what they're trying to prove. They assume that similarity equals ancestry, and therefore a lot of similar traits will be found within a baramin. But a baramin is a group, a holobaramin, in their words. A holobaramin is a group that is defined by ancestry. Defined by a group of organisms that are related to each other. But a group of organisms that are related to each other have a bunch of similarities. How do we know they're related to each other? Because they have similarities. How do we know that the similarities are because they're related? Why are they homologous, not homoplagious similarities? Because they're related to each other. Why are they related to each other? Because they have homologies. Why do they have homologies? Because they're related to each other. Circular, 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 circular. It's a giant circle. The entirety of statistical baraminology is a giant circle, and this is the foundation for it. This paragraph right here. This paragraph on page 350, if you're trying to find it. 30 years we've gone down this hamster wheel and accomplished nothing. If anything, we've undermined our own methodology. And when anybody dares to bring it up, they get plastered. When most of the similarities between two organisms are homoplagious similarities, then they are separated by a phyletic discontinuity. When a vast majority of the similarities between two organisms are homologies, then they can be considered part of the same monobaramin. How do we know they are homologies? Here we go. Next paragraph. Distinguishing homoplagious and homologous similarities, however, is not an easy task. In macroevolutionary theory, homoplasies are thought to be the result of independent convergent evolution. Convergent evolution is considered an unlikely phenomenon, especially if the feature is not strongly adaptive. Therefore, in contrary to creation theory, macroevolutionary theory claims that homoplagious similarity should be an uncommon feature of the biological world. For the purpose of minimizing error, then, conventional biologists automatically declare any similarity between organisms to be homologous in nature until suspected or proven otherwise. To make things worse, evolutionary systematics, which until the last couple decades was the conventional biosystematic method of the last century and a half, has prevented the recognition of much homoplagious similarity. Evolutionary systematics seeks to classify organisms in a way which reflects their phylogeny. By definition, homoplagious similarity does not indicate relationship, so homoplasies are simply ignored by evolutionary systematists. Homoplagious similarities were thought for many years to be non-data and were consistently unreported. Under the reign of evolutionary systematics, the impression was gained that homoplagious similarity is a very rare feature of life, just as macroevolutionary theory predicted. Yet, since homoplagious similarity would have been systematically ignored, even if it were present in common, is the rarity of homoplasia a real characteristic of life, or merely an artifact of the biosystematic method? Notice what he's not done. He's not told me how to tell a homology from a homoplasy. I don't think he can. I'll be honest with you, I don't think he can. But we'll continue on. And by the way, he's making good points about the evolutionary biosystematic model, but the problem is he's grabbing onto parts of it. The parts that are the problem. In the mid-20th century, cladistics was developed. Although not so intended, cladistics for the first time allowed the systematic identification of homoplasia similarity. Whereas the evolutionary systematists considered only homologous characters, cladists considered any shared derived character, even if those characters are homoplasias. A byproduct of the popularity of cladistics is that an increasing number of similarities formerly assumed to be homologous have been reinterpreted as homoplasias. It is suggested that cladistics methods can be used by baraminologists to locate homoplasies and determine their frequency. When no cladogram can be constructed for a group of organisms which eliminates homoplasy, then it can be concluded that homoplasia similarities exist. When a large number of characters is examined cladistically in a polybaramitic group of organisms, a young Earth creationist would expect that homoplasies would be unavoidable and frequent between organisms of different holobaramins. Conversely, for the organisms in the group which are part of the same holobaramin, it should be possible to construct a cladogram with few or no homoplasies. How do we know something's a homoplasy versus a homology? How do we know? It's assumed. It's hardwired. A similarity that results from ancestry is a homology. A similarity that does not result from ancestry is a homoplasy. So how do we know it's homologous? Because they share ancestry. How do we know they share ancestry? Because they have homologous traits. How do we know they have homologous traits? Because they share ancestry. How do we know that they share ancestry? Because they share homologous traits. And round and round and round and round and round the hamster wheel we go! This doesn't work. It can't work. It's self-referential. It's a giant circle, and we've been spinning on it for 30 years. I don't know exactly how you get out of this if you want to try and use homologies versus homoplasies. I don't think you can. And bringing in cladistics, this cladistic piece into classification, into baraminology, has ruined it. Because we now make the exact same assumptions that the evolutionists do in classification. A bunch of similarities? Yep, there's a homologies. Similarities that are not related? Yeah, they're homoplasies. How do we know? Well, because these two organisms aren't related. Therefore, these are homoplasias. How do we know they're not related? Because they have a bunch of homoplasies. How do we know they're homoplasies? Because they're not related. How do we know these two organisms are related? Well, they have a bunch of homologous traits. How do we know they're homologous traits? Well, because these two organisms are related. How do we know they're related? Because we have two homologous traits. Again, around that hamster wheel. 30 years and nobody caught this. I get that Kurt Wise is really popular because he's got a degree from Harvard. And he was a student under Stephen Jay Gould. I get all that. But this is 30 years in a hamster wheel. You'd think somebody, other than me, would have picked up on the hamster wheel. There's a lot of smart people in creationism. The problem is, the statistical baraminologists have been allowed to run wild. No one's ever checked on their work. It's never happened. Never been done. Until very recently. And the moment that it was done, they went full ad hom. Full ad hom. Cladists rarely find it possible to construct their cladograms without the use of a computer. They have designed a number of computer programs which can take the data from a character matrix and construct cladograms from them. Baraminologists should design their own computer programs which take the data from that same character matrix and construct baraminograms from them. The program will be designed to identify phyletic discontinuities according to the frequency of homoplasy. It would thus group organisms according to apobaraminic groups. The larger number of characters, the closer the apobaramins would be to monobaramins. The output of the programs, the so-called baraminogram, would include traditional cladograms for members of each identified apobaramin, as well as a list of the minimum number of homoplasies which justify each phyletic discontinuity identified. This is what was done with statistical baraminology, and it's a dumpster fire. You cannot tell a homoplasy from a homology just by looking at it. You have to assume something about ancestry. And that makes this system worthless. It's worthless. And statistical baraminology is based on this set of paragraphs right here. Frequency of homoplasy. That's pretty much the basis for statistical baraminology. And it directly has a cladistics background. Kurt Wise encouraged it to borrow from cladistics. So when the certain members of the young Earth creationist community come out and say, well, it doesn't have anything to do with cladistics, yes, it does. Yes, it does. It's right here, in black and white. It's right here. Read the paper. Now, I'm assuming that these individuals who are making this claim are making it in good faith. Probably some of them are, at least. But they're wrong. They're just wrong. Statistical baraminology is a cladistics-based method. Admittedly, it borrows more from phonetics than it does cladistics, and we can talk about phonetics another time. But it is partially based on the ideas of Kurt Wise, which come directly out of cladistics. The ideas of homologies and homoplasies. And those need to be abandoned, because they just don't work. According to the young Earth creation model, we're now in a different set here, flood-generated diversity bottlenecks. According to the young Earth creation model, the history of life's diversity has encountered at least one significant mass extinction, namely at the time of the flood. The diversity and abundance of the bios at the creation is unknown, but it is reasonable to assume that the 16 centuries or so after creation and before the flood, organismal abundance may have increased at a more or less consistent rate. It is more likely that the abundance increased logarithmically. It may have increased and leveled off as does bacterial abundance within a petri dish containing limited resources. If intrabaramenic speciation was a common phenomenon, then diversity also increased through the antediluvian period, and may also have done so geometrically. The flood, however, introduced a major extinction into the history of life. Post-flood land-animal diversity dropped down to the number of baramins. The diversity decimation in the marine realm may have been comparable. Since God purposed to save all the land baramins through the flood, it is reasonable to assume that he also intended all the aquatic baramins to survive the flood as well. It is likely that most of the intrabaraminic diversity was decimated by the flood, just as it was on the land. Large animal abundance dropped to 2 or 7 per species. Land animal abundance dropped to 2 or 7 per species. I think he means per kind there, or per baramin. Or unless he's referring to each individual baramin as one species, which the created heterozygosity model might not go with, but I'm not entirely sure why he says species there. He could be meaning one. He could mean that what came off the ark was a single species. That could be what he means. Although the abundance decimation in the marine realm was probably not nearly so severe, the number of marine fossils indicates that it was nevertheless extensive. And by the way, the vast majority of fossils are marine organisms. After the organismal abundance once again increased, probably in a logarithmic fashion, after the flood, that is, with a different, perhaps more varied post-flood climate and topography, organismal diversity may also have increased rapidly. However, it is likely that the organisms seen after the flood were rather different than those before the flood. The abundance bottleneck of the flood would allow for rapid allelic fixation, genetic drift, in the small post-flood populations. This, and the fact that the population which survived the flood may have carried less than the complete pre-flood gene pool, i.e. the founder effect, would cause post-flood populations to differ from the same baramins populations before the flood. The different climate and topography after the flood may well have encouraged a different type of interbaraminic diversity through natural selection than that which characterized the pre-flood world. Thus, the post-flood intrabaraminic morphotypes, species, etc., are likely to be different than those of the pre-flood period. This means that the flood-deposited fossil species would be expected to differ from the species of the present. Yes and no. I would expect there to be some differences, but genetics is somewhat determinative. So it is likely that a lot of the same traits popped up again post-flood. They may have been organized a little differently and shuffled around a little bit, but the kinds would not have morphed very much, most likely, I suspect. But Wise is a paleo guy. He's not a gene guy, so I'm not blaming him too much for that. If this is true, then all or nearly all species in flood sediments are likely to be different than modern species. I don't think so. No, I don't think that's correct, but okay. At some higher taxonomic level, however, the modern group will be recognizable in the fossil record. The taxonomic level where the groups go from non-modern to modern should approximate the holobaramin of land animals and birds. Depending upon how extensive the decimation of sea creatures were, the same reasoning could be used to approximate many of life's holobaramins. Alright, so now we're into the discussion section, which lasts for roughly 3 pages, and then we get to the conclusion. Having fun yet? Oh geez, 50 minutes already. Oh boy. Discussion. For baraminology to adequately function as an arm of the young Earth creation model, it is necessary that it be incorporated into the larger framework of the model. That means that it will be necessary to associate the terminology of baraminology with both the terminology of evolutionary biology, conventional creation biology, and the scripture. An initial attempt at this is included here. It seems necessary to introduce or reintroduce two terms in order to adequately bridge the gap and reduce potential confusion: the archibaramin and the baramin. The archibaramin. For each biological kind, Hebrew min, the originally created individuals comprise that kind's archibaramin. Monobaramins, holobaramins, polybaramins, and apobaramins of discontinuity systematics and baraminology are made up only of organisms for which we have physical evidence, either living or fossil. In contrast, it is possible that we have very little, if any, non-literary physical evidence of any archibaramin member. It is very unlikely that any archibaramin member survived to the present, and relatively few have survived to the time of the flood to be buried in its sediments. Evidences would include fossils made before the flood and not redeposited by the flood, e.g. pre-Vendian fossils, and peat plant debris, and archaeobaramin members still alive at the time of the flood but buried in flood sediments, e.g. some long-lived conifers similar to bristlecone pines. Potentially the Woolwami pine here. That's a possibility. The nature of the archibaramin as a concept is thus fundamentally different than that of the mono, holo, poly, and apobaramins. Rather than being restricted entirely to known organisms united on the basis of reproducible criterion, those indicating common ancestry, the membership and nature of the archibaramins is a theoretical construct and must be largely inferred and may never be reconstructed with certainty. Okay, yeah, that's true. The archibaramin is pretty much theoretical. I'm not entirely sure what good it does for us, but hopefully Wise will explain that. He's going to give us some more details here. Simplifying assumptions which are made about the archibaramins are listed as follows. The archibaramin was completely bounded by genetic constraints. Since the archibaramins of Genesis were constrained by divine command to thereafter always breed true, it is assumed that they were provided with some sort of internally defined genetic constraint which bounded them from morphological change in any direction. They were thus prevented from crossing with non-members and presumably from assuming any morphology outside these bounds. Although genetic constraints may have made particular intra-archibaramic hybridizations impossible, no genetic constraints completely divided any archibaramin. If they existed, intra-archibaramic genetic barriers only partially divided archibaramins. Thus, it would have been possible to find some particular series of matings which could make gene flow possible between any given archibaramin member and any other. Each of these matings might have produced reproductively viable offspring as well. Okay, that's solid. That's solid. Although gene flow was theoretically possible among all members of an archibaramin, intra-archibaramic gene flow may actually have been incomplete. If archibaramin membership was at all large, it is unreasonable to assume that all members bred with all other members of the archibaramin. Again, that's true too. And particularly if you think about the created organisms as populations rather than pairs, this makes it even more sense. 4. Archibaramin represents only a very small fraction of the morphological space defined by its genetic constraints. Because even a small region of multidimensional morphological space has a theoretically infinite variety of possible morphologies, and Earth with limited resources seems to make it unlikely that the full complement of morphologies were present in the archibaramin. It also seems reasonable that divine providence would provide the descendants of these organisms with the ability to adapt to conditions not yet realized. That seems like a precursor to Jeanson's created heterozygosity model. 5. The genetic constraints of the archibaramin defined a region of morphological space which did not overlap the morphological space of any other archibaramin. Now here, I am somewhat skeptical. And because I don't really buy the whole 3D morphospace thing. I think Ramine's concept of this is better, where it's a single line of continuous ancestry. But again, I don't know. You'd need a statistical program to model Ramine's ideas, and I don't know that that's ever been done. I'd like to see it be done, but get some statistician to help with that. If somebody's very good in statistics, by the way, if somebody's a statistician or a computer programmer and knows how to do this stuff, and you want to reach out to me, feel free. We can co-author a paper and try and work on some of this stuff. The baramin. Baraminology accepts the baramin as the created kind, Hebrew min, repeatedly referred to in Genesis chapter 1. Baraminology's reintroduction of Frank Marsh's term is with a slightly different meaning than that understood by Marsh. The baramin is here defined as the archibaramin and all its descendants. I don't know that I like that. As with the Archibaraman, the Baraman is a theoretical construct, for it is not restricted to known organisms, united by reproducible criteria of common ancestry. The Baraman includes its own Archibaraman, which is itself a theoretical concept, all known descendants of the Archibaraman, and all unknown Archibaraman descendants. Just as with the Archibaraman, the full characterization of the Baraman is known only by inference, without complete certainty. There are some things that can be known about the Baraman, and they are a consequence of features of the Archibaraman. They are as follows, and numbered according to the numbered features of the Archibaraman listed above, upon which they are based. So, number 1. What can be known about a Baraman? Number 1. The Baraman is completely bounded by genetic constraints, i.e. Baramans are apobaramans. Since the genetic constraints bounding an Archibaraman are thought to be internally defined and heritable, the Baraman is considered to be completely bounded by the same constraints. Thus, inter- or extra-Baramanic evolution is not possible. Okay, so that's what he means by evolution. He means change within a kind. Okay. I'm not a fan of that use of the word. I think that's unnecessarily confusing. But, okay. 2. Complete intra-Baramanic genetic barriers can develop over time. It seems possible for genetic constraints to both increase in magnitude and even arise where there were none before. It is also possible that intra-Baramanic extinction might prevent gene flow between two parts of the same Baraman. Scripture does not indicate that the Baraman members were always required to be able to successfully hybridize with all other members. It is thus conceivable that genetic and other constraints might arise to completely and permanently divide a Baraman into two or more gene pools. Thus, whereas successful hybridization with a reproductively viable offspring might be possible among all members of the Archibaraman, it will become increasingly less likely through time. Because of this, we are required to broaden the definition of successful hybridization to mean complete hybridization, regardless of the physical or reproductive fitness of the offspring. And that's true, by the way. Organisms that may have once been able to breed together may not be able to do so anymore. That is possible, and that is something that needs to be considered when discussing baraminology. There may be some more paper reading on that at some point. The Baraman might contain more than one holobarama. This is number 3. I.e., Baramans are not necessarily holobaraminic. Since gene flow within the Archibaraman was incomplete and relatively little time has elapsed since creation, it is conceivable that different portions of the same Archibaraman and their descendants remain genetically separate through time. This would be a true phyletic discontinuity, which is historically and not genetically constrained. Such Baramans would not be holobaraminic. Under these circumstances, the assumptions behind the hybridization and variation criteria of discontinuity systematics would be invalid, and would risk incorrectly identifying polybaraminic groups as holobaramans. So, in other words, let me read that again just to make sure we understand that. Since gene flow within the Archibaraman was incomplete and relatively little time has elapsed since creation, it is conceivable that different portions of the same Archibaraman and their descendants remain genetically separate through time. This would be a true phyletic discontinuity, which is historically and not genetically constrained. Such Baramans would not be holobaraminic. Under these circumstances, the assumptions behind the hybridization and variation criteria of discontinuity systematics would be invalid, and would risk incorrectly identifying polybaraminic groups as holobaramans. Okay, I don't believe that, because of the flood. You just talked about the flood bottleneck. I don't think it's very likely for members of the same Archibaraman to have remained genetically separate through time. Certainly not anything that was on the Ark. And anything that was not on the Ark is unlikely. The flood was a catastrophe of epic proportions. It wiped out almost the entirety of life on Earth. Very, very few proportionally organisms survived the flood. And you want me to believe that members of the same Archibaraman after the flood didn't interbreed? I'm sorry, I just don't buy that. I don't buy that. I don't think that's a valid. I don't think that's a valid comparison. I really don't. And I do think, therefore, that the Baraman is likely to contain only one holobarama. Further additive similarity criterion will be needed to minimize this difficulty. It will also be useful to acknowledge that groups are more likely to be incorrectly classified would be those with reduced gene flow. Holobaraminic classification can remain more tentative for those groups. All in all, however, this kind of error should have little detrimental effect upon creationist classification, because the resultant holobaramans will actually be closer to baramans. If better means can be found for identifying true monobaraminic relationship, then the criterion we already have can be used to unite holobaramans into baramans. Because it has little negative effect on creationist taxonomy, and we lack criteria to say otherwise, and its effects were minimized in the population bottleneck of the flood, it is reasonable for baraminology to assume that all phyletic discontinuities are due to genetic barriers and not historical constraints. I think Wise is making a good point at the end there. I think he's kind of saying what I just said. It's likely that everything is constrained by genetic barriers and not historical constraints that do not enable the genes to overlap. I think that's probably likely. Within a baramin, this is number 4. Within a baramin, there is considerable room for microevolutionary change, including speciation. Change of allele frequencies by genetic drift, founder effect, natural selection, etc. Allelic recombination, including the entire organ or species package, as well as unexpressed genetic material, mutations, chromosomal aberrations, and speciation are observed, real phenomena. Each of these operates completely within the confines of a baramin. With the exception of mutations and chromosomal aberrations, each of these is non-creative. That is, they produce no new genetic material, they simply combine genetic material in novel ways. In the case of mutations and chromosomal aberrations, there is no known case of an unadulterated benefit to the organism. It is likely that all such errors in replication and division are either neutral or deleterious to the organism. I'm going to temper that just a little bit by saying that they might be situationally beneficial. But overall, they're detrimental. That would be my minor caveat to what Wise just said. Number 5. The genetic constraints to the baramin define a region of morphological space which does not overlap with any other baramin's potential morphological space. A consequence of this feature of the archibaramin, a baramin is here assumed to be morphologically distinguishable from other baramins by an interbaraminic morphological gap. Now, this is great in theory, but the problem is you can't see this in practice. How do you surround a baramin with discontinuity? You'd have to display the entire baramin in 3D space and display other baramins around it and see whether or not they overlap. And the likelihood of this working is really small. And if you were to do something like this, it would be entirely, at least with the 3D software that's currently being used, it would be entirely subjective because you can spin it in any direction you want. It's not locked at all. I can make a 3D multidimensional scaling plot say anything I want it to say. So this one I'm kind of skeptical of here. Baraminology focuses on identifying hollow baramins. By focusing on organisms that are known and not hypothetical organisms, this process avoids the kind of disagreements and confusion that have classically plagued creationists as they discuss the concept of the biblical kind. Once hollow baramins are identified, then separate discussion can follow about methods of intra- and interbaraminic classification and taxonomy. Simultaneously, an increased understanding of hollow baramins will augment our understanding of the baramins. This will allow another distinct series of discussion through the nature of the archibaramin, the baramin, and the baraminic constraints. It is thought that baraminology will extract the creationist from the traditional mire of confusing terminology and allow him to more properly reclassify organisms, speculate about genetic constraints to change, and generally to better understand the biological creation. In a sense, I think this is true, but it's also been dangerous in other ways. Statistical baraminology, we're about to go into the conclusion here. Statistical baraminology, which arises from this paper, has largely been an unmitigated dumpster fire. It is statistical baraminology that has led to Todd Wood saying that there's multiple human species. It's statistical baraminology that has led to absurd things like humans clumping with chimpanzees and hyenas clumping with cats. It is statistical baraminology that has done this. And I think the reason being, the underlying reason is the homology argument. They do not understand. They either do not grasp, and I can't believe that Wise, who's a Harvard grad, doesn't understand this. Like, maybe Harvard education had gone down by the time Wise got there. I don't know, but like Wise is a Harvard grad, and his last name's Wise. He should be smart, right? I'm sorry, that was a bad pun. But he should be, with his Harvard degree, and he was a graduate student for one of the greatest evolutionary scientists of his time, Stephen Jay Gould. You'd think he would have understood enough of phylogenetics to realize that homology and homoplasia are circular terms. And yet he wants us to use them. I don't understand why he wanted us to use them, and why they were, of course, implemented. The ideas, at least, were, of course, implemented into statistical baraminology, which has led to a raging dumpster fire. We'll read his conclusion. Creation theory maintains that there are many real phyletic discontinuities among the Earth's biota. Current taxonomic methods do not reflect this claim because they lack any means of recognizing or identifying such discontinuities, let alone characterizing them. In order to make this evidence of creation available, there is a serious need for creation biologists to create, adopt, and employ a reproducible method of flagging phyletic discontinuities. With such discontinuities identified, the classification of the Earth's biota will then be in need of serious reconsideration. Although the classification within phyletic continuous groups need not change, the classification of such groups into larger groups is in desperate need of revision. And yet, continuity and the absence of continuity to the point of discontinuity is what statistical baraminology looks for. They've done the exact opposite of what Wise wanted them to do. And he just rolls along with it. Does he not see that? I don't know. And this is not meant to be a personal attack on Wise. He's a smart guy. And by the way, and just a side note here, in case anybody who thinks that this is a personal attack on Wise, he has probably the single most powerful testimony of anybody I've ever heard. It's available somewhere on YouTube. You can find it if you search for it long enough. I might even be able to find it myself and put it in the video description. So I'm not doubting Kurt Wise's salvation. I'm not doubting he's a creationist. That's not what I'm doing here. But I disagree very strongly with some of his conclusions, and I don't understand how he doesn't see the same problems that I see. And no, I will not shut up because he has a Harvard degree and I don't. Doesn't work that way. Credentialed experts don't get a pass. And he's not a biology guy. He's a paleo guy. I'm a biology guy. I have the ability to ask these questions and try to understand why it is that he can't see the big problems that I see. It is suggested that baraminology provides the tool necessary for the identification of true phyletic discontinuities, and thus the basis for a creationist taxonomic revision. With all the criteria of discontinuity systematics, the continuity criteria of successful hybridization, known natural and experimental variation, and unambiguous lineage, and the discontinuity criterion of the radical failure of the continuity criterion, and further young Earth creation model-dependent membership criterion, cladistically deprived homoplasy frequency, flood-generated diversity bottlenecks, and molecular typology studies aided by biblical criteria. Baraminology is the most efficient means available of identifying and characterizing true phyletic discontinuities. I don't think it does that. It doesn't do that at all, as a matter of fact. There is still much work that needs to be done in fleshing out the criteria already presented. Young Earth creation biologists are encouraged to critique, refine, and if necessary reject the proposed criteria. Even more importantly, they are encouraged to introduce new, better criteria. The more valid criteria that are employed, the more successful and rapid will be the accumulation of phyletic discontinuity evidence of creation. It will also be necessary to decide upon the best method of naming holobaramins, and then for classifying holobaramins into successfully higher groups. It is hoped that baraminology can be improved where it will be effective in meeting the classification needs of the young Earth creation biologist. And then he has several pages of notes on his paper where he is explaining words and so on. And of course, it only has 15 citations, which really isn't that many. He only has 5 citations. Take that back. He only has 5 citations and 15 notes, it looks like. 5 citations really is not that many. This is the paper that baraminology is based on. 5 citations. Whew. And then they have the trees, obviously, and then there's some discussion with people, with Harold Coffin in particular contributing, Wayne Frere as well, and Richard Lumsden contributed. I do believe all 3 of them are dead at this point. I'm not entirely sure, which is unfortunate. He does give us, however, the neo-creationist orchard model, which I am thankful for. That was the model I was taught in college, and it was very helpful to me in understanding what was going on. So these model, that was very helpful. This paper alone was worth the read just for those. I can hopefully have those put up on screen when I discuss them. Right? Final thoughts on this paper. Mixed bag, good and bad. Baraminology as a pursuit is worthwhile. Some of the stuff that Wise says is good. Some of the stuff that Wise says is bad. Your mileage may vary. Some people really like Kurt Wise. Some people really don't like Kurt Wise. I'm kind of neutral on Kurt Wise. I've never met Kurt Wise, but I think he's caused some serious problems within the creationist movement because of his insistence on cladistics and homology and so on in his baraminological methods. And the fact that they were adapted into something that is a 3-headed monster, effectively. All right, that's all we've got for this particular podcast. Let me know what your thoughts are. Ask me if you want a particular paper read. Let me know if you want me to talk about a particular subject. I will take requests on that score. Feel free to tune in next week. We'll probably have a video out sometime around then. And I look forward to seeing you all next time.